Hi everyone, and if you can't tell already, I love this toilet bowl background. I think it's funny. Anyways, hello and welcome to Chapter 8, Part 1D about hemoglobin. Now, last video, we talked about the general structure of hemoglobin that you have learned so far, how it is found in large quantities in red blood cells. There are many of these molecules in red blood cells. They are polypeptides. Specifically globular protein made out of 2 alpha globin chains and 2 beta globin chains, and they can bind oxygen to form oxyhemoglobin in the lungs. Now, today we're going to talk about hemoglobin in its entirety. What is hemoglobin for? There are two things. Number one is to transport oxygen to the cells. Number two is also to transport carbon dioxide of some sort back to the lungs and expel that carbon dioxide out of our body. So hemoglobin does two things. Number one, transport oxygen. Number two, transport carbon dioxide. But how does it do that? Now, before we go into that, let's talk about something called the hemoglobin dissociation curve. The hemoglobin dissociation curve is on a graph that is the percentage saturation of hemoglobin against partial pressure of oxygen. What do these two terms mean? Percentage saturation of hemoglobin is actually how much oxygen is bound to that hemoglobin. If the hemoglobin is fully bound with oxygen, then it's 100% saturated. Okay. And partial pressure of oxygen is concentration of oxygen actually. But you know, oxygen is not in liquid form here. Oxygen is in gaseous form. And therefore, partial pressure will be a more accurate way, more proper way of talking about concentration of oxygen. Yeah? So we can see here that the curve is, of course, increasing. Okay, but it's not directly perpendicular. It has an S shape here. And what does it show us? It shows us that as partial pressure of oxygen increases, percentage saturation of hemoglobin with oxygen increases. Now this makes sense, especially if you think about in the context of what hemoglobin does. Again, hemoglobin transports oxygen to the cells, right? It picks up oxygen at capillaries in the lungs, yeah, and then it sends it to capillaries in the cells, near the tissues, right? So at the capillaries in the lungs, you will expect high partial pressure of oxygen because oxygen supply is high. And therefore you have a hemoglobin that is very highly saturated with oxygen found at the capillaries in lungs. Make sense? Now in tissues, however, at the respiring tissues, the capillaries near there, oxygen is in high demand because those tissues will take in oxygen in order to perform aerobic respiration, right? To produce ATP in the mitochondria. So partial pressure of oxygen is low near the respiring tissues. And you expect the hemoglobin in the capillaries near those tissues to release oxygen and therefore be less saturated with oxygen. So it makes sense. Now, the only thing that is not answered here is why is it a S-shaped curve? Why is it not a straight line? Now, it's an S-shaped curve because hemoglobin, little typo there, exhibits cooperative binding or allosteric effects. And what happens is, as oxygen binds, this increases the affinity of hemoglobin for more oxygen. This means that, hey, when one oxygen binds, it will increase the affinity, which is a degree of attraction of hemoglobin for oxygen, which means the first one is quite hard to bind, but the second and third oxygen molecule will be easier. So let's look at this curve and think about it that way. So at low partial pressure of oxygen, so at this point of the curve, think of the first oxygen combining with the hand. Yeah? So it's still not that steep. And what happens is this changes the hemoglobin shape. And this causes the hemoglobin to increase in affinity for more oxygen. So the binding of the second and third oxygen molecule is easier than the first because the hemoglobin has changed shape, right? And therefore, a small increase in partial pressure, so you just increase a little bit, a higher increase in percentage saturation, which results in a slightly steeper curve on this side. And of course, at high partial pressure of oxygen here, hem groups are fully occupied, and therefore hemoglobin is fully saturated, and therefore the curve sort of levels off. So you can see from here that, hey, at first it's not so steep, then it became steeper, and then it levels off. Yes, that is why the curve is in this shape. What does cooperative binding and allostatic effects mean exactly? Well, you don't need to explain it usually, just throw the word at them in the mark scheme. But if you want to know, cooperative binding really talks about how oxygen binds and helps the second and third oxygen binds by cooperatively binding. Allosteric is because it causes it to change shape. So it's similar to the idea of the elasticity you have learned in enzymes, but it's not exactly the same here because hemoglobin is not an enzyme. I repeat, hemoglobin is not an enzyme. It is a molecule. So that is the hemoglobin dissociation curve. We have answered what it shows and why it is S-shaped. Now, the hemoglobin dissociation curve has its importance. And why is it important exactly? Now, this is a bit of a repetition, but here we go again. It's because in the lungs, oxygen partial, PO2, let's deal with that, are higher, right? And this means that's because there's oxygen's high supply and you can bind oxygen there, and therefore partial pressure is high, right? However, PO2 in respiring tissues are lower. So when you think about it, as blood travels from capillaries in lungs to tissues, in capillaries and then back to the heart, and then back to arteries and capillaries again, you expect a small decrease in partial pressure to lead to a large decrease in percentage saturation. So just as the hemoglobin detects a small change in partial pressure of oxygen will be released. This allows more oxygen to dissociate from HB, which is short for hemoglobin here. And affinity of HB to oxygen would decrease at low partial pressure of oxygen. Now, why do we say that the affinity of hemoglobin to oxygen decreases at low partial pressure of oxygen? It's because there are also other factors in play when it comes to affinity. Number one, there is partial pressure of oxygen, which we talked about just now. At high partial pressure of oxygen, it increases affinity of hemoglobin to O2. Yeah, more oxygen, more binding. But there are two things here, pH and PCO2, partial pressure of CO2, that has the opposite effect. At higher partial pressures of both these things, it decreases the affinity of HB to O2. This results in what we call the Bohr effect. Now, when it decreases in affinity, the curve would shift to the right, resulting in this second curve here. So this curve, purple color, is low in partial pressure of carbon dioxide, so like usual. But here, when there's a higher partial pressure of carbon dioxide, the curve is shifted to the right. Now, pH is not shown here, but you can assume that pH is also high when there is high partial of CO2, and you'll see why later on. Let's talk a bit more about the Bohr effect here. It's named after this guy, Christian Bohr, which is completely not boring, and he came up with this really cool theory, where the affinity, this is where the affinity of hemoglobin to oxygen is affected by PCO2, as we have talked about just now. What happens is high PCO2 decreases the affinity of hemoglobin to oxygen, and therefore a higher partial pressure of oxygen is needed to meet the same percentage saturation. So let's verify this by looking at the curve and why it shifts to the right. So analyzing the sentence, right, if it is the same percentage saturation, let's say around 60 here, For the low carbon dioxide concentration, you will need around 3 point something partial pressure of oxygen. But you see here at the high CO2 concentration, to reach the same percentage saturation, which is like around 60%, you would need 4 point something, almost 5 partial pressure, K pascals, partial pressure of oxygen. Okay, so that's why when you have to remember when the curve shifts right, this means that it has decreased affinity of Hb to O2. The reverse is true. If the curve shifts to the left, this means the affinity of Hb to O2 is increased. Okay, again, I'll say it again because it's very, very useful. If the curve shifts right, if the curve is to the right but normal, this means the affinity has decreased. This is a very important point. Okay, so how is the Bohr effect important and how is it helping like your understanding of the circulatory system? Bohr effect actually increases the dissociation of Haemoglobin in actively respiring tissues. So in actively respiring tissues where there's low partial pressure of oxygen, right, it will not only release some, it will release a lot more because of the high partial pressure of CO2. Now, if you don't understand what I just said, never mind, let's go through this again step by step, one by one. Lung tissues have higher partial pressure of oxygen and lower partial pressure of carbon dioxide. I'll just stick to PO2 and PCO2, it's just much easier, right? So lung tissues, more oxygen, less CO2. However, respiring tissues have lower PO2 and higher PCO2. More carbon dioxide because in respiration, oxygen is absorbed and carbon dioxide is released from those cells, right? So if you look at these curves, let's all look at that first. Let's look at these curves. There's two lines here. And you can see at different sections correspond to different parts of your body. Right, so when the lung capillaries have a higher partial pressure of oxygen and a lower PCO2, it belongs on this side of the curve, right, on this particular section. Because here, partial pressure of oxygen is high, and that curve is for the low PCO2. Active tissues, on the other hand, right, is this section of the curve, because this is the area where partial pressure of oxygen is low, yeah, and this is also the line that has high partial pressure of carbon dioxide. So different sections of the curve would correspond to different areas with different PCO2 and PO2. Okay, so seeing this curve, you realize that, hey, oxygen dissociates more readily from hemoglobin at respiring tissues because not only it has low oxygen, less oxygen, there's also a lot more carbon dioxide. And more oxygen will be released to meet the demand for aerobic respiration. So this is why the Bohr effect is important. Your tissues, your cells, which depend on oxygen, will get it readily because of the high partial pressure of carbon dioxide. But the question remains, how in the world does high partial pressure of carbon dioxide decrease the affinity of hemoglobin to oxygen anyway? Like, I know it decreases, but how? So this chemical equation here is how. And I recommend that you really memorize it and take it to heart. Okay, so what happens is this. CO2 from the respiring tissue, a side product, the product of aerobic respiration, diffuses out from the respiring tissue into the blood plasma, into the red blood cells. Remember, the tissue is separate from the blood vessels, right? So it diffuses from the tissue, blood plasma to red blood cells. Now, in the red blood cells, there is this enzyme called carbonic anhydrase in the cytoplasm. This enzyme, carbonic anhydrase, ASE enzyme, globular protein, everything you know what enzyme implies here, this enzyme converts CO2 to H2O to form carbonic acid, which is H2CO3 here. You can write H2CO3 in exams, no problem. Now, what happens is that this maintains a steep concentration gradient for diffusions of carbon dioxide from tissues to blood. This means that the CO2 won't diffuse until equilibrium. There's no such thing because it's immediately converted into another thing, so that CO2 is always high in the tissues and low in the blood. Okay, so carbonic anhydrase plays a part in this. And then carbonic acids, as all acids will, spontaneously would dissociate into hydrogen carbonic ions and hydrogen ions. Okay, so this is why we say that when carbon dioxide partial pressure is high, so PCO2 is high, pH will be high too because hydrogen ions are produced here. Now, but they don't stay that way. Something happens to hydrogen carbonic ion and something happens to hydrogen ion. We haven't answered the question, how does it affect the affinity yet, right? So here we go. So the hydrogen carbonic ion, HCO2 minus, diffuses from the red blood cells and goes into blood plasma. Okay, so it comes out of the cell, hangs on the plasma, still in the vessels though, and they sort of hang out there and get transported to the lungs via the vessels. Now, hydrogen ions, however, combine with hemoglobin. How crazy is that? They combine with hemoglobin to form something we call hemoglobinic acid, which is a mouthful, so I'll just call it HHB. But you need to remember this term, hemoglobinic acid. Now, HHB formation is because HB actually has higher affinity for hydrogen ions than oxygen. It binds to hydrogen ions more readily than oxygen. Besides that, hydrogen ions, besides that, hemoglobinic acid or HHB also prevents pH from decreasing. You know, we know the pH is low, but we don't want it to be too low. If not, it will denature the enzymes inside the red blood cells. So you don't want that. So HHB also acts as a buffer. Now, back to that point about hemoglobin having a higher affinity of hydrogen. If it does have a higher affinity for hydrogen, this means that hemoglobin will bind H and not bind oxygen. So hemoglobin releases that oxygen, and this oxygen obviously diffuses into the blood plasma and then diffuses out of the capillaries and gets supplied to the respiring cells. Oh, there you go. So yeah, that's what happens to the dissociated carbonic acid right here. So this is just a diagram of what just happened so far. CO2 in the plasma, yeah, gets diffused in carbon dioxide and water, H2CO3, okay, and then dissociation, and then these processes. But what is important here is the formation of HHB. Because without the formation of HHB, I mean, let's do it the right way around. With the formation of HHB, you realize that this answers our question. The high partial pressure of carbon dioxide decreases the affinity of hemoglobin to oxygen by the formation of HHB itself. As a result, oxygen is released and goes into blood plasma and gets supplied to those respiratory tissue. But as a side note, and also the important part of the syllabus, we need to talk about CO2. Now, what happens with HCO3 minus in the plasma? Now, of course, again, it's going to be all the carbon dioxide in your body. You want to get rid of it, and it has to go back to your heart and gets pumped into the lungs so that it gets oxygen again, and carbon dioxide has to get out of your body, right? So it doesn't stay in this form forever. CO2 is transported though in this form only 85% of the time. There's two more ways you need to learn about how CO2 is transported and then expelled from the lungs. So let's do this. So 85% of the time again is hydrogen carbonate ions. CO2 becomes hydrogen carbonate ions. And the whole process is the same as we just talked about just now, blah, blah. 5% of the time, CO2 actually dissolves in blood plasma and remains as CO2 molecules. Yeah. So that's dissolved and that can be transported. How about the remaining 10% of the time? And this is crazy, but the remaining 10% of the time is the CO2 combining directly to the hemoglobin. 10% of the time, carbon dioxide binds to the terminal amine of HB, and because it's a terminal amine, as in the amine group of the protein, yeah, this forms carba-amino hemoglobin. Carb as in carbon dioxide, amino as in the amino terminal, and hemoglobin. Carba-amino hemoglobin. 85% has hydrogen carbon ions, 5% dissolves with blood plasma, and 10% of time carba-amino hemoglobin. Insane. So this happens at the respiring tissues, right? When the partial pressure of carbon dioxide is high. Now, of course, as it moves to the lungs, the conditions will change. In the lungs, there is less carbon dioxide and there's a lot more oxygen. And therefore, CO2 will diffuse down the concentration gradient. And the processes are all reversed, including HHB releasing its hydrogen ions and binds oxygen to form oxyhemoglobin, because at this part, again, high PO2 is present. Now, what happens to carbon dioxide? Let's talk about it a little bit. Right. Number one, hydrogen carbon ions does the opposite. It diffuses back into red blood cells. It was previously in the plasma. Diffuses back to red blood cells, combines with the hydrogen, which got released from the hemoglobin, and converts back to carbonic acid, which is then converted back to CO2 and H2O. The same reaction is catalyzed by the same enzyme, carbonic anhydrase. Yeah? That is the 85%. How about the 5%? Obviously, the dissolved CO2 in the blood plasma, again, which is diffused down the concentration gradient into the alveoli. Number 3, 10%, the CO2 from carba-amino hemoglobin. This one will do the same, diffuse into alveoli, dissociate from hemoglobin, and get into the alveolus. We breathe out, CO2 gets expelled from the lungs. So everything in reverse, okay? So that's transport of carbon dioxide to lungs and transport of oxygen as well.